Okay, so as I said, today we will discuss the extraction, purification, and the analysis of nucleic acids. So that'll cover the half of chapter 6 and the whole chapter 7. Okay, and then the first one that we want to discuss is the extraction of the DNA. So the extraction of DNA is so important because in recent years, the advance in molecular diagnostic method provide fast and accurate detection and identification of clinical sample and the microorganism. And these techniques have been developed particularly that use the DNA or RNA, all right, and then try to analyze the DNA and RNA to get the information. So that is the reason why the DNA extraction is so important, okay, and recently in the recent years, especially the past, the last 10 years, it become extremely important. And so as you may heard a lot, one is the PCR, a real-time PCR, and then also the next generation sequencing. All these techniques require the extraction and purification of the nucleic acid. So that's why the DNA extraction is so important, and DNA preparation. And then the DNA extraction, actually, for the sample that we collect for analysis, DNA extraction is the first step in the laboratory procedure. Okay, and this because the DNA, as I mentioned, many molecular applications require the DNA. All right, so that would be the first step. And so the DNA extraction is important that we need to get a high yield and high quality DNA from any sample that we collect. Okay, and of course that the DNA quality and quantity depend on the technical requirement, time efficiency, and also cost effectiveness. That's all these are under consideration. And so we can choose a particular technique to extract the DNA. And then for those DNA that we want to purify, okay, we have to go through a 3 standard procedure. All right. So the first step is to collect the sample. Collect the sample can be the cells. All right. Collect the cells, and then once you collect the cells, then you have to break the cell because cells, they have cell membrane, and some plant or the microorganism, they have a cell wall. So you have to break the cell wall. You have to lyse the cell because the DNA is inside the cell, all right? And then for eukaryotic cell, the DNA is inside the nucleus. So that's why the first step, you collect the cell. And then the second step is to lyse the cell, release the cell extrait. And then the third step is to purify the DNA and the nucleic acid from those cell extrait. That means you remove the protein, carbohydrate, lipid from your specimen, so from your sample, so that you get the pure nucleic acid. Okay, and so remember, so in the next few slides, all right, don't get confused. The first step, we collect the cell, then we will lyse the cell, then we will purify the nucleic acid, all right? And then for the different specimen, there are different approach. So we can have a whole blood sample, we have dry blood spots, non-invasive human DNA isolation, or the DNA isolation from the microorganism. All right, that's these 4 different topics that we're going to discuss. Then the first one is the extraction, DNA extraction from the whole blood sample, okay? So as we know that the DNA obtained from patient, the whole blood sample is a key element for molecular diagnosis. And the isolation of a genomic DNA is the first step in running the molecular diagnostic assay. So it's essential to obtain a high purified genomic DNA from the sample, all right? And the whole blood, all right? Whole blood, that's the sample we collect, right? And then, so the whole blood can be routinely collected for the plasma and the peripheral blood mononuclear cell isolation, and using the centrifugation and the density gradient medium, all right, to separate the different cells. And then when you collect the blood, you have to collect the blood in the presence of anticoagulants. So what are the coagulants? Like EDTA and the citrate. These are the anticoagulants. So you collect the blood in the presence of EDTA or the citrate, right? So you have the blood sample like the, you have blood sample, and then you collect the blood, and then you mix with FICO. You basically you have a FICO in the bottom, and then you layer the blood on top of FICO, and so you will have two phases. Remember, the first step is always to collect the cells. So this step will help us to collect the cells. All right. So you basically you lay you add the blood over the FICO and then apply the centrifugation. After the centrifugation, basically that gives you 4 different phases. So you will have the plasma on the top layer, and then we have white blood cell below the plasma, and then in between the white blood cell and the red blood cell, that'll be the FICO. All right. So that's the centrifugation and also density gradient. All right, we use a FICO as a density gradient median. All right, and then after centrifugation, then you separate into these 4 different parts. And normally we collect the plasma and the white blood cell to perform the DNA purification. Okay, although sometimes nowadays people also can retrieve the DNA from the red blood cell, but the amount of DNA is minimum, is not much. Mostly is from the plasma and the white blood cell. Okay. So basically, for example, for the white blood cell, so the platelets that can be separated from the mononuclear cell by centrifugation through a fetal bovine serum cushion gradient. All right. And then after the centrifugation, the supernatant containing the platelet is discarded, and then the peripheral blood mononuclear cell can be collected for the DNA purification. Okay, so that's how you purify the DNA from the blood. And then these are the 2 quick genomic DNA extraction methods. Okay, and these 2 methods, they are not involved the FICO density gradient technique. And one is the proteinase K, and the other one is a phenol. And of course, the proteinase K. Proteinase K basically it's the enzyme that can degrade the protein. Okay, so in this protocol, you can see that the whole blood mixed with a trace EDTA, SDS, magnesium chloride, and proteinase K in the presence of high salt. And then you have the overnight digestion at 37 degrees. Okay, and after this digestion, the next step is the DNA purification, which we will discuss in the next section. Okay, again here also, after you separate this different layer, then you collect this the mononuclear cell, all right, mononuclear cell from the white blood cell, and that mononuclear cell will be subject to the DNA purification. Here, I only present you the first step, how to collect the cells, all right? Collect the cell and cell extract for the DNA purification. And here again, the proteinase K, all right, try to break down the cell and then release cell extract. And the DNA, of course, subject to the further purification. And then for the phenol method, the whole blood, you mix with the saturated phenol. And then shake in 4 hours at room temperature. Also try to break the cell, okay, and remove those contaminants like the protein. Okay, so these are 2 quick extraction methods. One is the proteinase K, and the other one is the phenol. Okay, for the blood. And then the dry blood spot preparation. So generate the genomic DNA extract from the whole blood has been widely used for diagnosis. Okay, but the dry blood spots, DBS, and filter paper have emerged as a convenient way to collect and transfer specimens for genomic DNA extraction in rural area. Okay, because in rural area you might not have the effective cold storage, and also the transport is difficult, so that you can use the DBS method. And the many advantage to use the DBS for genomic DNA preparation. So basically, you can do it in the room temperature, because normally when you collect the blood, and the blood must be stored at 4 degrees and processed as soon as possible to obtain the maximum DNA yield and the quality. And in contrast, the collected blood spot, DBS, okay, that can be dried at the room temperature and shipped without refrigeration. And furthermore, the DBS can be collected from the finger sticks, okay, or the heel sticks, like for the infant and the young children. So that this blood collection procedure causes a minimum risk, okay, to the participant. Okay. And another advantage is DBS can be kept in a long-term storage facility for years before the DNA extraction without a significant loss in quality. For example, it has been shown that the DNA extracted from blood spot stored for up to 25 years can also be successfully retrieved the genomic DNA for the PCR reaction. Okay, so the dry blood, and also it's a simple collection procedure and stability of the DNA in the blood spot. The DBS allow the collection to be handled by adult without requiring the participants to visit the medical facility. Okay, so these are the advantage to use the DBS to prepare the DNA. Okay, and then of course when you have the DNA on the filter paper, then to collect the DNA, then you have to release the DNA from the DBS. And so you punch out the DBS from the filter paper and treat with the 5.5% softening or the PBS overnight 4 degrees. Then the filter paper can be subject to the Chelax or Instagene metric method to purify the DNA, which we will discuss next. Okay, so that's the DBS preparation. And here are the two different methods to prepare the DNA from those DNA released from the dry blood spot. Okay, so we have this Chelax 100 or Instagene matrix. If you soak the sample in the saponin, so basically, for example, you place the filter paper in 0.5% saponin and 4 degrees overnight, and then you remove the saponin and add 1 milliliter of the PBS buffer, and then incubate at 4 degrees for 30 minutes. And then you remove the PBS and place the filter paper in the 100 microliter 5% Chelax 100. So you incubate 100 degrees for 8 minutes, and then you apply the centrifugation, and then so the DNA will be released to the supernatant. So that's why you collect the supernatant and store at the minus 20 degrees if the X-ray is not used promptly. So basically, so this is the step that you collect the DNA. All right, you break the cell, collect the DNA. So this is a Chelax 100, and then we have instagene matrix, the two different methods that you can collect the DNA. For the non-invasive human DNA isolation, you can get it from the hair or urine. And here this slide tell you how to get the DNA from the hair. All right, from the hair, you can use the simple alkaline lysis buffer. So basically, you collect the hair with root. Remember, the DNA present in the root. So it's like this, the root, the hair follicle. You need to collect the whole hair, right, from this end to the head here, right, with the root, because this is the area that you can get the DNA. And then you incubate 95 degrees for 10 minutes in the sodium hydroxide buffer. Okay, so this is the alkaline lysis buffer. And then the supernatant subject to the DNA purification. So this step here is just to release the DNA into the supernatant. Then you collect the supernatant for the next step, to purify the DNA. And then another method is the chemical digestion method using the DTT. So you have the hair with the root, incubate 2 hours at 56 degrees, all right? And then with the buffer, you see the proteinase K, all right? And we have DTT. Did you see the DTT? Okay, DTT is the basically is the chemical. Okay, that can like a beta-mercaptoethanol. They are the reducing agent, and the reducing agent they can break the disulfur bond in the protein. All right, they can break the disulfur bond in the protein, and the proteinase K is to degrade the protein. Okay, so you incubate the hair with the tris EDTA, sodium chloride, SDS, DTT, and proteinase K. And then you mix it and incubate 60 degrees for 2 hours or until the hairs dissolve completely. All right, and then the DNA will be released into the supernatant. Okay, and the supernatant is subject to the further purification. So this is the method to collect the DNA from the hair. And then from the saliva, all right, the saliva that you can swab, okay, you can have a buccal swab sample, right? And then suspend the lysis buffer. The lysis buffer with tris EDTA, SDS, and proteinase K. So these basically can break the protein, okay? And then you incubate a sample, 56 degrees, 1 to 3 hours. And then you can extract the DNA from the solution. Okay, so that's the DNA collected from the saliva. And then this is the DNA collected from the urine sample. Okay, so basically you collect the urine, all right, you invert the specimen cup to create the homogeneous suspension of cell. And then you apply you invert the tube several times to mix it and then apply the centrifugation. After the centrifugation, then the supernatant is removed. And then a dry pellet containing cell can be chilled at 20 degrees, minus 20 degrees for 15 minutes. And then you can add the lysis buffer. Okay, again, the lysis buffer, tris EDTA, SDS, and the proteinase K. Then 56 degrees for 2 hours to release the DNA to the solution. Okay, and then you can purify and concentrate the DNA from the sample. So this is the technique to purify the DNA from the urine sample. Okay. And then to prepare the DNA from the microorganism. So So as we know, the bacteria and the fungi, they have a cell wall that must be broken to release the nucleic acid. Okay. Usually both are chemical method, which means that you lyse the cell by expose this cell to the chemical agent that can affect the integrity of the cell barrier. And the other method is the physical method. The physical method, you use the physical disruption, okay, like a sonicator. Use a sonicator. You have a probe to break the cell, or you can use the ultrasonic sonicator. Use the ultrasound to break the cell. That's a physical disruption. Okay. For the chemical method, that's use the chemical reagent. All right. So you use a detergent cell lysis. It's a milder and easier alternative to the physical disruption of cell membrane. Okay. Basically, the detergent breaks the lipid barrier surrounding cell by the solubilizing protein and disrupting lipid, protein, and protein-lipid interactions. And non-ionic and the literal ionic detergent are milder and less denaturing than ionic detergent. So the non-ionic detergent are used to solubilize the membrane protein. The, one of the chemical is called CHAPS, 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 which is a literal ionic detergent, and another one is called Triton X. It's also a non-ionic detergent and commonly used to solubilize the membrane protein. And the ionic detergent, like SDS, they are strong solubilized agent. They cannot denature the protein and destroy the protein activity and the function. And for bacteria, so we have to break the cell wall, all right, and release the cell X-ray. A commonly used chemical is called lysozyme. Lysozyme is very efficient for the breaking down the cell wall of gram-positive bacteria because their cell wall have a high portion of phosphorus. The cell wall, the bacteria cell wall, because they are gram-positive. They have the peptidoglycan. Right? When you learn the microbiology, you learn that the cell wall for those gram-positive bacteria, they have a lot of peptidoglycan, so that you use a lysozyme to digest the peptidoglycan. And the gram-negative bacteria, they are less susceptible to lysozyme, but the combination of lysozyme and EDTA can effectively break the cell wall and membrane. And the mechanical disruption method that can be used to disrupt the gram-positive bacteria cell and the spores efficiently. Okay, that's the mechanical disruption. And then the fungal cells are difficult to disrupt. Okay, because the cell wall can form the capsule or resistant spores. And liticase, all right, liticase, also known as the zymolase, can be used to digest the cell wall of the fungal cells. Cell can also be broken by the alkaline chemical detergent. The fungal genomic DNA can also be extracted with the CTAB, right? CTAB is the cetyl trimethyl ammonium bromide method. So basically, the CTAB mixed with the 3-CDTA hyso buffer and the conidal suspension, followed by the incubation at 65 degrees for 1 hour, so that this method can break the fungal cell wall very efficiently. So we have lysozyme for bacteria and the liticase, zamolase for the fungi. Okay, that's DNA preparation from the microorganism, and the Chilix 100. Okay, Chilix 100. Chilix 100 is a chelating resin. They have the high affinity for the polyvalent metal ion, and also can be used to isolate fungi DNA quickly and efficiently. Why there's no response?
Can you repeat the question?
Oh, nobody listen. Okay, so the Chelex 100, okay, is very important because it's a chelating resin that has been used very commonly, okay, to isolate the DNA because it has a high affinity for the polyvalent metal ion, and also can be used to isolate the fungi DNA quickly, efficiently. It can be used to release the DNA from cell by a boiling treatment. At the same time, protecting the DNA from the boiling effect with resin beads. So typically the conidal suspension from the sample is extracted by adding the 5% of Chelex 100 resin, and then you incubate the mixture at 90 degrees for 30 minutes, and followed by the centrifugation, and then you transfer the supernatant, all right, and then add another Chelex, and then you incubate again, and then the supernatant. After the centrifugation, the supernatant is collected for the DNA purification. So this method is quick to obtain the DNA from spores. Okay, so that's Chelex 100.
Professor, are you talking about endospores, the heat-resistant structures?
Yeah, for the fungal spore.
How would you not denature the DNA if you're using a high temperature?
So that's why you use Chelex and also resin B. They can protect the DNA.
So it's
and
like a binding?
90 degree will not break the DNA because when you, for example, the DNA, the phosphodiester bond, the covalent bond is very strong. 90 degree, 100 degree will not be able to
It's
break
not
the
hot
DNA.
enough to break the bonds.
No,
Okay.
no. Okay, it's Chelex 100 method. Okay, so you see, you have Chelex, right, with the resin, and then you have supernatant. So you mix your DNA with the Chelex and solution, and then the DNA will be released to the supernatant. Okay, so you can use this, for example, hair tissue or saliva, and then, or you can do the microorganism, the fungal, to collect the fungal DNA as well. Use the Chelex. And this is just to show the DNA quality evaluated by the PCR reaction, PCR amplification. And we have these two different type of methods. One, from lane 1 to lane 6, use the Chelex 100 to prepare the DNA. And then 7 to 12 use this CTAB method to prepare the DNA from the fungi. And then you can see the Chelex 100 give you a better quality and quantity after the purification and the amplification. Okay, so until now, we discussed how to obtain the cell and then break the cell to release the DNA, all right, into the solution. Once the DNA is released into the solution, the next step is to purify the DNA, all right? So you have to purify the DNA from the crude cell extract. And then this step involves the removal of a protein, carbohydrate, lipid, and cell debris. And we have different techniques that can be used to remove protein, carbohydrate, lipid, and cell debris. Okay, so that you can purify your nucleic acid. Okay, and then of course the first step, the first method is organic method. So the organic method uses the organic chemicals, right, including the phenochloroform to purify the DNA. Or you can use the inorganic method, so the detergent like EDTA, acetic acid, and salt. Or you can use the salty phase, like use the beads and column to isolate the DNA. Okay, for the first one is the organic purification. So for the organic purification, normally use a phenochloroform extraction. Okay, that's commonly used, right, to purify the DNA. The phenochloroform extraction. So what you need to do is you have the cell extract, and then you mix the cell extract with the phenol. Right, you mix it well, and then apply the centrifugation. After the centrifugation, so that we have two layers. So after the centrifugation, you have the organic layers, or you have an aqueous layer. All right. The organic layer will be the phenol, and then the original cell extract will be the top layer, which is aqueous layer. But using the phenol, basically the phenol will bring all the protein into the interface, so that the function, the role of the phenol during the DNA purification is to remove the protein from your sample. Okay. And although the phenol, they can denature the protein rapidly, it does not completely inhibit the ribonuclease activity, like the RNase activity. If you are going to collect the RNA from the sample, normally you will use a mixture of phenochloroform isoamyl alcohol, right, to purify the nucleic acid, because this mixture can inhibit the RNase activity during the purification process. And also you can also add the proteinase K and RNase, all right, to inhibit the RNase activity and also to degrade the protein. So that's the first step, you use the phenol. And then the next step, after you separate these two layers, then you remove this top layer to another new tube, and then you mix it with the chloroform. All right, so you can mix this with chloroform, mix it, and then subject to the centrifugation. All right, so after the centrifugation, and then again you will get two layers, and the upper layer will be aqueous layer, the bottom layer will be a chloroform. And the reason you use a chloroform after phenol is because chloroform can remove the all the phenol residue from the DNA sample. So that's why you, after the phenol extraction, then you add the chloroform to further purify the sample. All right. And then after that, the upper aqueous DNA containing layer is now pure and can be further concentrated. Okay, so a high concentration of salt, like a 0.3 molar of sodium acetate, in the presence of 2.5 volume of 100% ethanol, or 1 volume of isopropanol at minus 20 degrees or below, and you can precipitate the DNA. And then DNA precipitate can be collected through the centrifugation. And then the excess salt can be rinsed with 70% of the ethanol and centrifuged to a discarded ethanol supernatant. And then the DNA pellets can be dissolved with tris EDTA solution for long-term storage. Okay, and sometimes the protein content is so great that only one single phenol extraction probably is not sufficient to completely purify the nucleic acid. And then you can perform several phenol extraction, one after the other, all right? So that you can remove the protein completely. But be careful that the more phenol extraction you perform, the more likely you might be able to break the DNA molecule into a small fragment. And the problem is when you break the DNA molecule into a small fragment, then when you perform the PCR reaction or NGS analysis, the result might not be reliable. Okay. And the other alternative is you can just add the proteinase K before the phenol extraction. Then you can remove those excess amount of protein. So that's the organic purification of DNA. And then for the inorganic method that involve the incubation of nuclei with only a proteinase K at 65 degrees. And then later on, the proteinase K can be inactivated by incubating the sample at 65 degrees for 2 hours. Okay, then you can inactivate the proteinase K. So the inorganic DNA isolation, you can do the proteinase K, all right? Or you can use the sorting out. That's another simple inorganic DNA isolation method. So you have a saturated sodium chloride. You use that to precipitate the protein. And then the DNA is purified from the supernatant by using the 1 to 2 volume of the minus 20 degrees ethanol. Then the pure DNA can be obtained. Okay, so that's the sorting out. That's two different methods for inorganic DNA isolation. And then for the solid phase nucleic acid purification, basically you can find these mainly the commercial extraction kit, or the commercially available DNA extraction kit. They use the solid phase nucleic acid purification because this allows a quick and efficient purification compared to the conventional method. Okay, so basically the solid phase system can absorb the nucleic acid in the extraction process. Okay, and basically the principle is the hydrogen bonding interaction with the hydrophilic matrix under the chaotropic conditions, the ionic exchanger under the aqueous conditions through an ionic exchanger, and the affinity and size exclusion mechanism. So these three different principles that you can use to isolate, purify the DNA. Okay, and also the solid phase purification normally performed by a spin column through the centrifugation. And this method can purify the nucleic acid rapidly. Okay, and you can use the silicon matrix, glass matricals, diatomaceous and an ionic exchange carrier. These are commonly used as a solid support in the solid phase extraction. So for example, the diatomaceous earth extraction. So the diatomaceous earth is known as the diatomite, has a silica content as high as 94%, can be used for filtration and in chromatography. It's useful for the purification of plasmid and the DNA by immobilizing the DNA onto its particle in the presence of a chemotherapy agent. A chemotherapy agent basically is a molecule that can disrupt the non-covalent bond in general. And the famous chemotherapy agent is a urine guanidine. And then the resulting diatomaceous earth-bound DNA can then be washed with an alcohol-containing buffer. And then the alcohol-containing buffer can then be discarded, and DNA is eluted out in low-salt buffer or the distilled water. And of course, the major step that involved in the solid phase, the first step is lyse the cell, and then you have the nucleic acid absorption, and then washing, and then elution. Okay, just like the diagram here, right? So you have the so for example, pegylated bacteria, then you have to lyse the bacteria. And then after you lyse the bacteria, then you run the cell X-ray through this column, right? And then the DNA will bind the filter, and then you can wash the filter, and later on you can elute the DNA using the low salt buffer. Okay. So basically, when you run the sample through the column, the nucleic acid will bind to the column under the high pH and the high salt condition, because that's a binding condition. All right, high pH and high salt concentration. And then when you try to elute the DNA, you use a low salt concentration, low salt solution to elute the DNA. All right, so that's the solid phase DNA isolation. The another one is the affinity extraction. That's the magnetic separation. It's a simple and efficient method, can be applied in the purification of nucleic acid. So very common that magnetic carrier with the immobilized affinity ligand, right? They have affinity to the nucleic acid. We can use that for the isolation process. And the magnetic particulate material basically are the beads, all right? The beads are the preferable as support in this isolation process because they have a larger binding capacity. The nucleic acid binding process can be assisted by the nucleic acid wrapping around the support. So in a certain concentration of the salt and the polyalkyl glycol, the magnetizable cellulose can bind to the nucleic acid, and small nucleic acid require higher salt concentration for strong binding to the magnetizable cellulose. Okay, so when it goes through this column, then it bind, and then later you can elute the DNA. And this method normally can be used to isolate protein as well. Okay, that's affinity extraction that we will discuss in next class. The anion exchange resin. This resin, basically, remember the DNA is negative charge, so you can have a resin as a positive charge. Okay, so the resin normally we use a DAE, all right, diacetyl amino acyl cellulose, DAE, all right, DAE, because the DAE is a positive charge on the resin surface, and then they can bind the negative charge, the nucleic acid. And this is the anion exchange resin, okay? So you have a defined silica beads with a large pore size, and then a hydrophilic surface coating and a high charge density. So these ion exchange resin, then cell extract goes through that, then the DNA bind here, all right, and later on you can elute it. And the DNA prepared from the FFP sample. So as we know, the fresh sample, okay, fresh frozen tissue sample are the best material for isolation of a DNA of high quality and quantity, but sometimes the storage of frozen tissue sample is expensive and time consuming. So for many retrospective studies, the formalin fixed and the paraffin embedded FFPE material is therefore the only available sample for the DNA analysis. And so that's why we discuss this here. You try to retrieve the DNA from this FFPE sample material. So we archive the biological sample from the patient as the FFPE block. And remember to make the FFPE block, the tissue first immersed for 2 to 3 days in formalin, and then you have the fixation and then the tissue block are dehydrated, okay, using the xylene and ethanol series and embedded in the paraffin wax. So these tissue block are therefore referring as the FAPE sample. Since the procedure of tissue fixation and the embedding has a profound effect on DNA, the challenge to obtain DNA of satisfactory quality and quantity depend strongly on the type of fixation used for the preservation. And here is a list of a preservative, okay, the fixative that you can use to fix the DNA. All right, the fixative you use is now will affect your downstream DNA quality and quantity. So like this here, the 10% buffer neutralized formalin is a good fixative, right? Because the DNA, when you retrieve, is good condition. And acetone also good, and then hope also good, right? So these are commonly used fixative and then can give a good quality and quantity of the DNA. A 10% formalin, 10% neutralized buffer, 10% formalin, that's the formaldehyde, okay? So this is formaldehyde. It's most widely used universal fixative. It can preserve wide range of tissue and tissue components. Here is just to give you the more detail about the mechanism of fixation by the formaldehyde, which is 10% formalin, okay? So we have to know that formaldehyde react reversibly with water to form a methylene glycol. So the 10% formalin, that'll be 3.7% of formaldehyde in water with 1% methanol. Okay, and in the initial process, you apply the alcohol fixation, followed by the cross-linking phase. The alcohol fixation basically causes dehydration in the process, hardening the tissue and the membrane. And the tissue are rapidly penetrated by the methylene glycol. So the actual covalent chemical fixation depends on the fraction of the formaldehyde forming a bond with the tissue. Okay, this formaldehyde forming a bond with the protein. For example, here is protein, and the formaldehyde comes from the dissociation of a methylene glycol. And this is the in the end you can see how they fix the protein. Okay, and then to retrieve the DNA, the first step is to deparaffinization. Okay, to remove those fixed exit, and the deparaffinization you use a xylene. Okay, and then washing in the descendant series of ethanol, and then protein digestion, and then DNA purification. So here is just to show you how to dewax. Okay, after dewax, and then again you lyse the cell, and after you lyse the cell, you release the DNA in the supernatant, then it's subject to the purification. And here is just to show the precipitation of the DNA, ethanol precipitation. So remember that the nucleic acid can be precipitated in the presence of salt, alcohol, ethanol, and the minus 20 degree or below. And so they can precipitate the DNA. All right. Once you precipitate the DNA, you remove the ethanol, then you can resuspend the DNA in the desired volume of the TE. All right, the 3-CDTA solution. So for long-term storage, and then you can determine a DNA concentration, which we will discuss in chapter 7. Okay, and then we continue. This is RNA preparation. It's basically very similar to the DNA preparation, except RNA is subject to the RNS degradation. Everywhere you will find RNS. So that's why when you prepare RNA, it has to be extremely careful. Okay, you have to use the strong denaturant to denature the RNS, so to inhibit the RNS activity. All right, so that you will not degrade the RNA. And RNA, as you learned from previous chapter, we have ribosomal RNA, messenger RNA, and transfer RNA. And the amount of messenger RNA is the less. All right, if your goal is to get a messenger RNA, then you have to be even extremely careful, because the RNS is heat stable, and also it can refold following heat denaturation, right? So that's why normally use the DPC, right, like 0.1% reagent that can inhibit the RNS activity. Or you can use the RNS inactivation. You can use a guanidine cytosine, which is a strong chemotropic denaturant. Or you can use the guanidine isocyanate, to capital ethanol, or DTT. So that you can add it to a extraction buffer, so to inactivate the RNS activity. Okay. And this is just the a commercially available system that you can collect the RNA from the blood. It's called a PEX gene system. So basically, you collect the blood, and then you add it into this PEX gene tube. And because the PEX gene tube contains a stabilized additive, so they can stabilize the RNA, right? Stabilize the RNA. And then here is just to show the comparison that if you collect the blood, through the EDTA packaging, and then after the 5 days, you see the ribosomal RNA, right? After 5 days, the EDTA cannot preserve well. See the packaging can preserve well, right? Okay, and this is the GITC column-based method. GITC, remember here, this here guanidine thiocyanate is the RNA inactivation reagent. Okay, so you use the GITC-based method to purify the RNA, right? So I include the cell lysis in the trizole. That's the trizole. That's the GITC inside the trizole. Okay, so they can stabilize the RNA, right? So this is the mixture of the GITC and phenylchlorophyll into the trizole. So you mix it, and then after the centrifugation, then you have several layers. All right. And then you have the supernatant, release the DNA to a supernatant, you add the propanol, high salt concentration, then you have the RNA pellet, right, after centrifugation. So you can wash the RNA pellet with the 70% ethanol, and then you can concentrate, dry it, right, concentrate. Right, that's GITC, and GITC stands for the guanidinium thiocyanate, all right? And this side will be the column. So basically you mix it with the trizole and then add the ethanol and then go through the column. And then the column, basically RNA will bind to the column. You wash the column and then elute it from the column, all right? And on the right side here, just the cell lysis plus lysis buffer and the ethanol, and go through the column, right? And then you collect the RNA. And this is just the comparison from these three different methods. So you have the trizole, tri-reagent, right? Or the trispin, that's the GITC with column or just column, all right? So the higher yield for GITC based method. This method is very similar to the phenylchlorophyll extraction. The difference is the instead of only phenol, they have the guanidinium cyanide, the GITC. So it's called trizole. Higher yield, okay, and but it take longer time, all right, demand more technique, and column based, quick, easy, but the yield might be low, right? And if you combine the GITC with column, quick and easy, but the RNA yield may be low, higher cost. And this is the GITC chlorophyll extraction, all right? So basically you have the guanidinium thiocyanate, which is a trizole, all right? The guanidinium cyanide is a calotropy agent, okay? They can degrade the protein, especially to inhibit the RNase activity, right? And then you have to perform this activity at low pH, all right? And then it mix, and then after the centrifugation, again, the phenol organic phase will be in the bottom with the lipid, and the interface will be DNA, aqueous phase will be RNA. So that's the special application of the GITC, different from the pure phenylchlorophyll extraction. And the spin column, so basically alternative to the GITC extraction, that will be this the column, spin column, okay? Basically the sample after the homogenization, okay? And then you pass through the spin column that can bind the nucleic acid. And then you can have the alcohol acetone add to the fluid, all right? And then after that, you can elute the RNA by using the water or tris EDTA, the spin column. And this will be a special application if you want to purify only the messenger RNA. The messenger RNA is important is because if you want to evaluate whether the gene expression level is changed, and then you have to use this method. So remember, in the eukaryotic cell, after the RNA is produced, they have to go through this maturation process. So you have to put the 5' cap and 3' poly A tail. So use this particular characteristic. Then you develop this technique, use to isolate the poly A directly. Okay. So basically, you have this affinity chromatography with the oligo-DT cellulose. And then when you go through this column, then those messenger RNA will bind to the column because they're oligo-DT beads, all right? And then you wash the bead and then elute the messenger RNA. And then another method to purify the messenger RNA is to remove the ribosomal RNA, okay? To eliminate the ribosomal RNA, there are many methods can be used. So for example, you can use a subtracted hybridization with ribosomal RNA specific probe, reverse transcription with ribosomal RNA specific primer, followed by RNSH digestion to degrade ribosomal RNA in the ribosomal RNA cDNA hybrid. Okay. So these are all different techniques that you can use to remove the ribosomal RNA and then preserve the messenger RNA. All right, so we finished the DNA and RNA purification. All right, and now we will watch some video. All right, about how to purify. So, for example, here DNA extraction from the blood. Okay.
Carried out in duplicate on a single blood sample. Please note that blood sample collection should always be performed by a phlebotomist. Before starting your DNA extraction, ensure that the area where this is being carried out is clean and has been decontaminated. This can be done by spraying the bench, the pets, and equipment to be used with freshly made.
This step is very important. Always you have to sanitize your bench.
Wipe away any residual bleach, followed by.
So for example, when you come to the lab next week, always remember you have to clean your bench with 10% bleach, all right? So each bench, we have the bleach on each bench. You have to sanitize the bench. And also, when you finish the experiment, before you leave, you also need to use the bleach again, all right?
Rinsing with clean water, and finally spraying with 70% ethanol. Gloves are to be worn at all times throughout the extraction process and change.
So remember, when you are in the lab next week, you need to wear a glove all the time.
Frequently, especially if coming into contact with other surfaces that might be contaminated. Also check that all equipment and consumables you are going to use are in place, and that the buffers in the kit have been prepared. For example, the appropriate volumes of ethanol have been added to buffers AW1 and AW2.
They use a kit, so that's why they have AW1, AW2.
Your heating block must have been set at 56 degrees centigrade and ready to use. Before commencing with DNA extraction, please ensure to familiarize yourself with the protocol. So you have prepared the area where you will be carrying out your extractions, and you will begin by labeling the microtubes with sample ID.
When you come in, remember all the experiment. When you perform the experiment, when you take the tube, you have to label the tube, all right? Label the tube with your initials so that we know that's yours, okay?
Using a permanent marker by inverting the tube approximately.
So this is blood.
To ensure you have a homogeneous sample. Vortex the proteinase K briefly, followed by a quick centrifugation step, approximately 10 seconds. Pipette 20 microliters of proteinase K into each microtube. Pipette 200 microliters of blood into each tube. Do this slowly as blood is viscous. Cap the blood sample and put to one side. Add 200 microliters of buffer AL to each sample. Close the tube and vortex for about 15 seconds, and this step will be repeated for all subsequent samples. Transfer the microtubes to the heating block that has been set at 56 degrees centigrade and incubate the samples for 10 minutes. At the end of the incubation period, remove the samples from the heating block and briefly centrifuge to remove any droplets from the lids. Add 200 microliters of absolute ethanol to each microtube and mix each sample by briefly pulse vortexing for about 15 seconds. Briefly centrifuge the microtubes. Prepare a mini spin column for each sample. Label your spin columns, ensuring that each spin column corresponds to the sample ID. We usually label the sides of the spin columns as well, in case the ID on the
So that's the column, spin column, okay?
Carefully transfer the mixture from the previous step to the spin column, taking care not to wet the rim of the spin column.
To here.
And this is repeated with all subsequent samples. Close spin column and centrifuge at 6000 G, that's about 8000 RPM, for 1 minute. At the end of the centrifugation period, all the filtrate should be in the collection tube. Transfer the spin columns, which now contain the DNA, to clean collection tubes and discard the collection tubes containing the lysate.
So discard those
Carefully open the spin
flow
column
through.
and add 500 microliters of AW1 buffer, taking care not to wet the rim. Remember to change your tip between each sample. Close the caps and return the samples to the centrifuge. And centrifuge 1 minute again at 6000 G, that is 8000 RPM. At the end of the centrifugation period, check that all the filtrate is in the collection tube. If there is still some in the spin column, just repeat that centrifugation step. Discard the collection tube and transfer the spin columns to clean collection tubes.
Right, yeah.
Carefully open the lid and add 500 microliters of AW2 without wetting the rim of the spin columns. Close the cap and centrifuge at full speed. That's 20,000 G, around 14,000 RPM, for 3 minutes. Discard the filtrate from the collection tube into a waste container, as the collection tube is going to be reused. Dry the rim of each collection tube on a paper towel and replace the spin column. Centrifuge at full speed for 1 minute. This ensures that all residual wash buffer and ethanol are removed, which could affect downstream processes. Label clean 1.5 microtubes, one for each sample, and transfer the spin columns to each corresponding microtube. The DNA that was on the spin column will be eluted from the column to the AE buffer. Add 200 microliters of AE buffer to each spin column, remembering to change tips between samples. Incubate for 1 minute at room temperature. Centrifuge for 1 minute at 6000 G, that's 8000 RPM. Re-elute the filtrate, transferring this back onto the spin column. Repeat the incubation and centrifugation steps. This repeat elution step optimizes the DNA yield for each sample.
Okay, so that's how you elute the DNA in the end, all right? So that's the DNA extraction from the blood. And then also here's another video about DNA purification. Yes, Marina.
I wanted to ask you a question about the PowerPoints. I noticed I can only access the videos like if I go to the recording. Like I can't physically play the video in the PowerPoint presentation. I'm not sure why. Like if I just open up the PowerPoint like in itself and I go to the videos, I can't just watch them. Like I have to go into Blackboard.
Right, you have to go to the recording, exactly, yes. You
Yeah.
can only watch the video through a recording, and the video is not embedded.
Oh, so I can't play it like in the PowerPoint. I
No.
wasn't sure if I was doing
No.
something wrong, and I wanted to ask
No,
you.
no, you did not do anything wrong.
Okay.
That's yes.
Yeah, because
Okay.
I like the videos. They're good.
Yeah, it's good, and you can watch it from the recording. This is plasmid DNA, so perhaps we will skip this one. The plasmid DNA will be a different approach that we will discuss later. All right, so we will skip this one, and then we will quickly watch this RNA. Total RNA extraction.
One protein and gloves to avoid any contact of reagents with the skin. Particular attention should be paid to putting on the gloves without touching the outer surfaces in order to prevent contact with residues of RNases from the operator's hands. At this stage, as the sample has not yet been added to the lysis buffer, the virus is still potentially infectious, and the operator may increase his safety by wearing.
So this is the video to show how to extract the RNA, virus RNA, okay? So that can apply to the, for example, the SARS-CoV-2, how to extract the RNA from SARS-CoV-2 as well.
A filter face mask. Extra care should be taken when working with suspected zoonotic pathogens. In diagnostic laboratories, RNA extraction is commonly performed using commercial kits. Commercial RNA extraction kits are generally based on the same principles and should be used following the manufacturer's instructions. In this video, RNA extraction using a glass fiber filter-based method will be presented. Begin the work session by marking the tubes with the corresponding sample identification number.
This step is the most important step, okay? You have to always label the tube so that you can identify which sample is in which tube, all right? If this step, you mislabel the tube or you skip this labeling, then you will make the whole procedure just meaningless because you don't know what is what sample, yeah.
The extraction procedure starts with the lysis of the tissue in order to...
So the first step always collect the cell or tissue and then lyse the cell or tissue to release the cell extract.
Free the nucleic acid from the cells. This is done by adding the sample to the lysis buffer provided in the kit, according to the proportions recommended by the manufacturer. This buffer contains reagents such as guanidine and Triton X that destroy cells, denature proteins, inactivated RNases, and create appropriate binding conditions that favor the absorption of RNA to the silica membrane. Dispense the required volume of lysis buffer into each tube, remembering that guanidine is harmful by inhalation, in contact with skin, and if swallowed. In some kits, the addition of ethanol is required to adjust RNA binding conditions. Addition of the sample is now required. Note that for each batch of samples, a negative control should be included by adding phosphate buffered saline to the lysis buffer instead of the sample. The correct amount of homogenized tissue under examination is now added to the lysis buffer. From now on, the operator can continue to work without the face mask, as the virus is no longer infectious. Vortex the solution in order to homogenize reagents and to avoid pelleting of any precipitate in the tube. Load the lysate onto a silica membrane column provided, which absorbs nucleic acids. Identify each column with the identification number of the sample. Dispense the lysate into the middle of the column, making sure that the filters are not touched by the tips in order to avoid breaking the glass fibers. Since this is a high-risk contamination procedure, it is strongly recommended that one tube is opened at a time and to change tips after each sample in order to avoid transfer of RNA by contaminated tips.
It's important that only open one tube at a time, and every time you have to change the tips, okay? Just to minimize the cross-contamination.
Remember to change gloves before leaving the hood. Centrifugation is now performed to allow absorption of nucleic acids to the filter. The duration and speed of the centrifugation are set according to the manufacturer's instructions. Now return to the hood and prepare new collection tubes into which the column will be placed for the next step of the procedure.
You see, she removed, after the centrifugation, everything goes through the filter and then down to the bottom in this tube, in each of these tubes. And now she removed this column, move to the new tube, remove the column, and then put the new tube. And you're going to discard this flow through, all right? It will keep it here because all the nucleic acid is in the column now.
Discard the flow through into the waste vessel. In certain kits, a desalting buffer is used to render DNA's digestion more effective. It is crucial to change tips in dispensing reagents from one sample to another in order to avoid contamination of the stock solution. To avoid this risk, it is strongly recommended that aliquots of the stock solution of each reagent are prepared. A second centrifugation is now necessary to remove the desalting buffer and to wash away any salt and proteins. The sample is now ready for DNA's digestion. During this step, total DNA present in the sample is degraded, and only the total RNA of the sample remains on the filter. The DNase reaction mixture is prepared according to the manufacturer's instructions in a single tube for all samples, and then dispensed into each single column. Incubation for 15 minutes at room temperature is necessary for the reaction. Note that this reagent may cause sensitization by inhalation and skin contact. Additional washing and drying steps are now performed using washing buffers specifically developed to remove salts, metabolites, and macromolecular cellular components from the RNA sample. Remember that all these washing reagents contain guanidine, which is harmful by inhalation, in contact with skin, and if swallowed.
The guanidine is to inactivate the RNase activity. Okay, remember.
After washing, RNA elution is now possible. Prepare nuclease-free collection tubes that have been properly identified in order to collect the RNA eluate and to store it. Place each column into the nuclease-free collection tube and discard the flow-through from the final wash into the correct waste vessel. Elution of RNA is done by adding RNase-free water. The water dissolves the low ionic binding between the silica membrane and the nucleic acid. After a final centrifugation, the RNA is eluted and the extraction is completed.
Okay, so that's the procedure to collect the virus RNA. Okay, so that'll be the end of the nucleic acid preparation, purification.